Welcome back to the pharmaphorum podcast. In this episode, I speak with Brian Fiske, Chief Scientific Officer of Mythic Therapeutics, about antibody-drug conjugates, or ADCs, and what's possible now, and what might be bettered, including reduced toxicity, in the future. Circling around the notion of patient-centered science and the importance of tackling eligibility, the conversation makes for some fascinating oncological insights. I do hope you'll agree. Tune in, let us know your comments, and as ever, thank you for listening. This is web editor Nicole Raleigh, and today I have with me Brian Fiske, Chief Scientific Officer of Mythic Therapeutics. Brian is an expert in cancer metabolism, and we'll be discussing ongoing trends in the antibody-drug conjugate, or ADC space, and developments in targeting and Mythic's work in this area. Welcome, Brian.
Thanks for having me, Nicole.
So, ADCs are designed to deliver chemotherapy directly to tumours. However, more than 95% of the dose often ends up in healthy tissues, decreasing efficacy and increasing toxicity. But Mythic, fueled by its fate control platform, develops self-navigating ADCs that precisely target cancer cells while sparing healthy ones, amplifying efficacy and minimizing side effects for patients. Before we delve further into ADCs, though, Brian, perhaps you could share with listeners a little bit more about yourself, about your journey, your passions, everything that led you to do what you do today with Mythic.
Absolutely. So by training, I'm a PhD in cancer biology. I completed my PhD in Matt van der Heiden's lab at MIT. Matt is certainly an expert in cancer metabolism, and I did a lot of work in that field with him during my PhD. Subsequently, after completing my PhD, I ended up at Flagship Ventures, which is now Flagship Pioneering. They're a early stage biotech-focused VC here in Boston, and had the role of working on starting a number of companies essentially off their balance sheet. So doing venture creation, and specifically started two companies, one in the cancer space and one in the intersection of rare disease and fertility. After Flagship, got very excited about starting my own thing. So started Mythic independently, and that's what I've been working on for the last 7 years. In terms of passion and excitement, I think there have been, maybe I'll talk a little bit about Mythic's values. I know we'll talk about Mythic later, but one of the things I was really excited to build about Mythic is sort of embodying what I see as Mythic's core values. One is patient-centered science. Technology is really exciting, but what really gets me excited is using technology to create a really great drug for patients. So at Mythic, we're much more excited about what that drug's going to be rather than necessarily building a technology. We know we need to use novel technology in order to make a great drug, and maybe make a great drug that others aren't building. We don't want to build me toos, but we're really excited about building great drugs for patients. The second is what we call built together, which means we're most excited about Mythic to work with people who will come in and build something and be entrepreneurial. So that's something comes from my personal excitement about working with people who are entrepreneurial and entrepreneurs. And then I'd say, lastly, another thing that's been really durable is a value we call fuel efficient, which means that we try to do more with the resources that we have, right? The idea there is, the way biotech works, at least at the early stages before you have a product, would be you go out, you raise a bunch of money, and you use that money to collect data, whether that's preclinical data or data in a clinical trial with patients. And wouldn't it be exciting if we could do twice as much with the resources that we have? So that's another thing that I get really excited about.
Brian, especially for sharing the values of Mythic. I think that makes it quite clear for listeners, the sort of foundational premise from which you do the work that you do, that we'll be discussing in a second. So if we turn to ADCs now, the topic today, an ASCO publication described ADCs as embodying a simple but elegant vision for cancer therapy, the delivery of a potent cytotoxic agent to tumor cells with minimal damage to normal cells, or what's so-called smart chemo. And the idea of targeted chemotherapy, for those unaware who are listening today, was conceptualized by the German scientist Paul Ehrlich over a century ago. And Ehrlich also coined the term chemotherapy, where he proposed to use chemicals to kill the pathogenic cells. However, the first ADC trials didn't get underway until the 1980s, and the first FDA-approved ADC came only in 2000. And pardon my pronunciation, but listeners might recognize the name gemtuzumab ozogamicin, used to treat acute myeloid leukemia. And the most successful application for solid tumors so far has been in breast cancer, with ADCs becoming the standard of care across traditional human epidermal growth factor receptor 2 or HER2+, hormone receptor plus, HR+, and triple negative disease subtypes. It's thought, however, that modifying the payload to use immune stimulating agents or combination therapies with immunotherapy and other effective targeted therapies may further extend the utility of these agents in the treatment of solid tumors. Indeed, it was also noted and published recently in Nature Review's Drug Discovery that ADCs are being increasingly used in combination with other agents, including as first-line cancer therapies. And as the technology to produce these complex therapeutics has matured, many more ADCs have been approved or are in late-phase clinical trials. So, Brian, I was wondering if you could comment on all this and discuss also the broadening scope of tumor indications for ADCs and the potential within combination therapies.
So I think the largest ambition of the entire ADC field would be to replace chemotherapy with something better. Everyone who's not a scientist, who's been affected by, or who is a scientist, regardless of whether you're a scientist or not, I guess, and has been affected by cancer knows, or has a family member affected by cancer, knows that when you think of chemotherapy, yes, you can think of getting better from cancer, but you also are thinking about how sick chemotherapy makes people, which is just really unfortunate. And so the premise of ADCs to be as or more effective than chemotherapy, but potentially have reduced side effects is really attractive, right? I think that's something that a lot of people can be inspired by, and certainly were inspired by in the ADC field. And I think where ADCs are now, you mentioned in HER2, you mentioned some of the earlier drugs like gemtuzumab. When ADCs work, they work really well. So if you're a cancer patient and you get informed that you're eligible for treatment with an ADC because of the particular type of cancer that you have, that's really great news. That should be really exciting that you're eligible for this treatment that's based on a chemo mechanism of action, but it's going to be much more specific, potentially more effective and safer. So that's where we are as a field right now. And we have a number of examples. So I think there's over 10 approved ADCs at this point. I think the issue is in achieving the larger vision of how do we just replace chemo in the future is how can we have eligibility for ADC occur for more patients, right? How can we have the experience of people understanding that they're eligible for an ADC happen to more patients? And the issue in solid tumors is that ADCs today really only work for patients or targets with very, very high levels of expression. ADCs work, they're really two major components. One is an antibody component, that's the A in ADCs. Then there's this chemotherapeutic payload, which is the D in ADCs, and a linkage between them. And they work because the antibody component can bind a target which is specific to tumor cells and not present at such high levels in normal cells, and then direct that chemotherapy towards that cancer cell. But correspondingly, the reason why ADCs are only working for very highly expressed targets and patients who express very high levels of the target on their tumor is because the more target that you can have on the tumor cell, the more ADC molecules it will bind and the more chemotherapy it will be delivering to itself, which kind of makes sense, right? If you have a targeted therapy, the more of the target that you have, the better that you'll do. The issue is that the number of targets that are super highly expressed like that, like the HER2s of the world, Nectin-4 is another example in bladder cancer that had some really great results in the last year. It's enfortumab, a drug from Seattle Genetics, now Pfizer. The number of targets that are like that, you can count on one hand. And the number of, outside of that, the number of patients, for example, the target of our lead drug, MET, only about 6% of patients with lung cancer express enough MET to be well treated by current ADCs. And so maybe to wrap it all together, we think at Mythic, a key problem for ADCs is how do you treat patients with lower levels of expression, maybe even intermediate levels of expression, so that we can achieve this vision of replacing chemotherapy with something better.
Thank you for that. So Discussing all this and coming towards what Mythic itself specifically does, what remains an issue here is toxicity and better understanding and management of those ADC-related toxicities, and how that'll be essential for further optimization. So I was wondering if you could expand on those toxicities and side effects and explain what Mythic is doing, basically.
Absolutely. So the platform that we built at Mythic, we call Fate Control. That to us means an understanding of how the targets of these ADCs, how they internalize into cancer cells, how they traffic throughout cancer cells. There are a lot of different compartments and areas within a cancer cell that a target can go, and maybe even how those targets recycle from cancer cells, how they go back out of the cell. And so a key part of that's the understanding that enables our platform. Our goal is to be able to use that understanding to design antibodies, again, the component that's responsible for delivering the chemotherapy, but as you mentioned at the very beginning, isn't very efficient at doing that. Only a small percentage of the payload actually gets to the tumor. We want to be able to increase the fraction that goes to the tumor where you want the chemotherapy to be delivered, and decrease the fraction that goes to normal tissue. And right now, that's pretty lopsided, the wrong direction, right? If you have 5% of the payload going to tumor cells and 95% going to, if it's not going to tumor cells, it's going to normal cells. And so, again, we use our Fate Control platform to deliver more to tumor, less to normal at the same time. And that's embodied in our lead drug, MYTX11, which is currently in a first-in-human trial in monotherapy for non-small cell lung cancer patients against this target called MET. And we're trying to show, yes, on one hand, that we can expand the patient population, but we're also potentially seeing things consistent with being able to deliver less to normal cells, because that drug, at least in our preclinical studies in animals, we've been able to show that we can deliver 3 to 5 times more payload to tumor cells, but we can also deliver at the same time 50% or less of that same payload to normal cells.
Thank you, Brian. And for listeners, that preclinical data was announced yesterday, was published in a journal of the American Association for Cancer Research regarding MYTX11. What I realize we've not spoken about, Brian, as well, is the potential for resistance to ADCs. The advent of ADCs for the treatment of metastatic cancers over the past decade has significantly improved outcomes across different solid tumors, as we've been discussing. Nevertheless, patients on these therapies can eventually experience disease progression because of resistance. Is that correct?
Beyond just a yes or no, yes, that's very true. Yes. I think it's unfortunately, as a field, I don't think anyone has a good understanding of why patients become resistant to ADCs. There are probably a number of good hypotheses as to why that happens. There's sort of individual cases here and there where you can point to something that's happened that could be related to becoming resistant to an ADC, but I think I haven't seen anything comprehensive about how resistance occurs. I also think there's very mixed data, maybe the indirect evidence even is mixed as to how that happens. But I do think it is something that hopefully we'll learn more about, and it's going to be important to learn more about, particularly as there are more ADCs, especially with regards to how we sequence therapy, right? So what if there are 2 ADCs against the same target, but with different payloads? This might happen, for example, in the HER2 space within HER2, right? And HER2 has HER2-directed antibody for mostly for breast cancer. It has a so-called topoisomerase inhibitor payload because it inhibits a protein called topoisomerase. That's the job of the chemo payload that it has. But there are other HER2 ADCs that are going to come along that have tubulin inhibitor payloads. So that's a different type of payload. So which one do you give first? That's probably one of the biggest questions. But are patients who relapse and progress on, an Enhertu, do they respond as well as a patient who's never seen as to Enhertu to a new HER2 ADC with a different payload? And all sorts of combinations in between. You could think about giving a patient 2 ADCs with different payload or with the same payload, but different against different targets. There are hundreds of ADCs in development. Many of them hopefully will prove that they are useful drugs, but hopefully we'll get more data and I think we'll want more insight into how resistance works in order to be able to sequence these therapies.
Interesting that you say there are hundreds in development, but there's only about 10 approved, did you say earlier?
Yep.
That's the nature of the game. So my final question was actually going to be what next for ADCs? I think we've approached it slightly in what you were saying there with understanding more about the whys and the wherefores for resistance. But is there anything you wanted to add or to summarize with, Brian, when it comes to that sort of horizon looking for antibody drug conjugates and indeed for Mythic?
Yeah, so I might go back to the thing we were talking about earlier, which is our mission at Mythic, right? I think Mythic is very unique in our focus on the antibody component of an ADC, right? Virtually all other companies in the space are focused on the linker payload component. And the linker payload component, the advancements in technology there over the past 30 years have been critical to getting where we are as a field. But I think the question is do we need something else? We've solved a set of problems. Do we need something else to solve the next set of problems? And I would argue that the next set of problems is really how do we get to these intermediate and lower expressing targets in patients? And so that's what we're solving at Mythic. We're solving in a very different way. That carries some risk to it, but I think it's also exciting to think that we could add another component to ADCs. And we can also take advantage of everything people are doing in the linker payload space. So fundamentally, our platform is payload agnostic. So we can deliver more to tumor cells and less to normal cells with any given payload. So I guess in terms of future of ADCs, I would love to see Mythic succeed. I'd also, to the extent there are innovations in the linker payload space that are going on, particularly maybe with respect to new payload classes that people haven't explored before, I think that'd be really exciting. I mean, one of the areas people are starting to get excited about are so-called degrader payloads that would go into a cell and degrade a certain protein that might be causing cancer. So that could be a really exciting area for Mythic. I mean, we're a small company. We're not going to build a lot of capabilities with degraders, but we could potentially think about partnering with a company that is and working together. Interestingly, part of the reason why existing payloads work is because they're so potent. And so the need for extreme potency is probably a real limiting factor with respect to new payloads. And that's exactly the problem that we're solving at Mythic by delivering more, right? You could potentially have a less potent payload by being able to deliver more of it. So those are some directions, that Mythic is involved in, maybe one that Mythic could be involved in, but, maybe we're thinking about less is I'd love to see people apply ADCs to spaces outside of oncology. So are there applications for ADCs in autoimmune disease, for example, with steroids? And there's some work going on there. Could we use the same sort of approach to deliver oligonucleotides? That's another thing people are involved in. So I think the idea of targeting a molecule or a class of molecules that already exist more specifically to tumor cells and less elsewhere, where they can cause toxicity is probably generally applicable. And I'd love to see more companies, broadening the scope of what we can tackle with the approach.
There's excitement and indeed there's hope too. It's been a pleasure.
Thanks, Nicole.
And so that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on iTunes, Spotify, Amazon Music, Podbean, and pretty much wherever you get your other podcasts. Just search for pharmaphorum and press subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us as well on X or Twitter and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.